welcome and thank you all for coming. And let me just say what this is and what the goal of it is really. So the idea of this, regardless of your vocabulary and if you knew what a charrette was, which I confess I did not know. But the idea is to get together talented people that we have here at the University of Rochester that have an interest in schizophrenia research and to start talking about reaching some goals together. And so in the discussions that John Foxe and I have had, what we thought would be good things to think about in terms of next goals after today are, for one thing, to start being a group, a psychosis research work group, really, that among other things would allow us to review each other's grant proposals and strengthen those prior to submission to funding agencies. We also would like to have some type of regular meeting, maybe once a month or every 2 months, where we have either a journal club type of discussion or discuss works in progress, or just grant ideas, and again, helping people kind of move forward. And ultimately, we'd like to get a schizophrenia or psychosis center grant here. And you know, what's exciting to me, and one of the reasons I came here to the University of Rochester really was because there's so much talent really in psychiatry and neuroscience and data management, data analysis, all kinds of other areas, and development, for example. So I think a really interesting proposal could be put together based on the strengths that everybody has. We just have to do it. So the point of this is to have as the first step in doing some of these things, and hopefully we'll have a center grant in the 2 to 3 years. So for today, I think the idea really was to have a few of us give some presentations on work that we're doing now, really to introduce ourselves and to kind of see what the possibilities might be for collaboration and for what type of center might we be able to put together and who might be able to run different cores and that type of thing. And then just to have a discussion about what people are interested in and where this might go. So I'm encouraged that so many people are on. So we need to start with the presentations, and I know Elizabeth Berry is on here. Elizabeth, can you say, I don't have in front of me the order of the presentations, but at least the first few of them, what the order is?
Yeah, we've got Julie Fudge, we've got Ed Lalor, we've got David Federer. There's no particular order. So, we've got Brian Keene, we've got Judy Thompson, and you and John.
Okay. Well, in the spirit of getting things over with, maybe I'll go first. Unless there's a loud objection, which I don't hear. So, okay. These are supposed to be, by the way, 5-minute presentations and with maybe a minute or two for questions afterwards. But again, the idea is just to stimulate thinking about the kind of work that we could do as a group. So I'm going to now attempt to share my screen. Hopefully this will work. So can people see this PowerPoint?
Yes, looks good, Steve.
Yeah, looks good.
All right. Well, so this is my opening slide. But this isn't really what I want to talk about here. So just to give a background about me, I just came to the university in January as Associate Chair for Research in Psychiatry. I had been at the U of R all through the 1990s, from mid-1990 to the very end of 1999. I started my career here in psychiatry. So it's good to be back. I got my PhD in 1989. I'm a clinical psychologist by background. I have a longstanding interest in schizophrenia research and treatment. I've run inpatient units, outpatient programs, mostly been a researcher in my career though. And for over 30 years now, believe it or not, I've been studying cognitive and perceptual impairments really in schizophrenia, mostly in vision, but again, other things as well. I wanted to show some of the work I've done here. This is my first data publication from 1992, which was on perceptual organization, or the ability to kind of integrate visual features into object representations quickly and automatically. And this was in people considered to be at risk for schizophrenia. This is something I started having an interest in graduate school. Within a few years, I was looking at the contribution of bottom-up, kind of obligatory stimulus grouping versus top-down effects on perceptual grouping based on what we expect to see, based on knowledge, based on context, and thinking about to what extent are the problems in schizophrenia that you see with organizing visual information. We also have audition as well. To what extent are those due to bottom-up versus top-down contributions, or what's the relative contribution of each one? And so where in the brain might these problems be happening? Within a few years, it became apparent pretty quickly that, of course, not all patients had these problems, but the ones that did tended to be ones who not only showed reduced organization of the visual field, but also reduced organization in other areas, such as loose associations in thinking, disorganized speech, inappropriate affect, so the emotion doesn't match the what they're thinking, even some disorganized movements. And so it seemed that these visual perceptual organization tasks could be good laboratory measures of a more widespread problem in just mental representation organization by context. And that would have implications then for clinical trials and other uses of these measures. Eventually, I became interested more in the biological basis of this, and in particular how it fit with the NMDA receptor hypofunction hypothesis, something I'm still interested in, and that's what this paper was about. Somewhere around 2004, 2005, I started doing fMRI studies of perception in schizophrenia, and looking at, again, what brain regions were affected and where was the earliest evidence of a problem. And also around that time got interested in, could these perceptual problems be changed either with practice or training or learning or something like this? So were they stable or not? And if not, how could they be changed? And what would happen if you changed them? This was a review paper I wrote with Brian Kane, who you'll be hearing from in a few minutes. He came to Rutgers as a postdoc, then as a faculty member, and now as a faculty member here in Rochester. I've also done some work with some consortia trying to optimize these measures for clinical trials, and also did some imaging work with these measures. And so these can really be used now in impaired populations. And most recently have gotten interested in computational modeling of some of the perceptual problems in schizophrenia. But currently, I'm not doing so much work in perceptual organization anymore, at least not independently. Brian Kane is doing some fantastic work that you'll hear about in that area. Most of my work these days is on the retina as a biomarker in neuropsychiatry, especially in schizophrenia. I want to say here that there's a lot of work in this area in neurology and neuro-ophthalmology, showing that changes in retinal function and structure, like thinning or loss of retinal tissue, is related to things like loss of brain volume, cognitive decline, illness progression, the number of disorders like MS and Alzheimer's and Parkinson's. But it's only been studied most recently in schizophrenia for the last few years. There are many benefits of studying these. They're generally quick techniques. They're non-invasive, or they can be non-invasive if you do them in a certain way. I noted here they're inexpensive with an asterisk. That means if you already have the equipment, it's very inexpensive. But as usual, it can be expensive to get the equipment initially. I wrote a review paper on this topic back in 2015. There were only a few studies done at that time. They were fairly consistent, though, in showing changes in schizophrenia. Now there are many more studies. This is one of mine from 2 years ago with a grad student. And just to show here, the typical waveform that you get from electroretinography, measuring kind of like an EEG of the retina, you get these different waveforms that are well known to reflect activity in different cell types with different types of activity and different neurotransmitter bases, et cetera. So there's a lot you can learn from this type of work. And of course, you do find changes in schizophrenia and the amplitudes and sometimes the latencies of these waveforms. I do want to say, though, that like with the psychophysical and fMRI studies, this is not something that's limited to the eye. You do find that these are essentially predictors or correlates of other types of problems in schizophrenia. So essentially, the retina is providing you with some proxy measures of the brain. They're all part of the central nervous system. In terms of retinal imaging, with optical coherence tomography, you can get these really nice images of retinal layers, and you do find thinning in schizophrenia, and fairly consistently in almost every study. In terms of current questions, one big one is, when in the course of illness do these changes happen? A person working with me, Adrian Lai, who's here, recently had a paper accepted showing that in first episode they're normal. So this seems to be an aspect of illness progression. But we don't know exactly when it starts. We don't know how much it's related to cortical thinning and cortical volume loss, or the extent to which the ERG changes really are proxies for neurotransmitter findings. In Parkinson's disease, for example, the extent of dopaminergic cell loss is clearly related to things like the ERG waveform changes, and L-DOPA will restore the function. But in schizophrenia, we don't really know what the consequences are. We don't really know how these changes affect visual perception in schizophrenia, whether it's real or causing hallucinations. We don't know how normal visual input contributes to the development of schizophrenia, although we do know that visual changes and visual impairments in childhood are very good predictors of later schizophrenia. And we also know, and this was reviewed here with Brian and myself, that people who are born blind almost never develop schizophrenia. And a number of studies have revealed that now. And so there's an interesting kind of side topic there. Finally, can these changes be remediated? This is some work, thing I've written about. This is a paper with two grad students of mine, and another one here with Judy Thompson, who completed a study of this. And we have a visual remediation clinical trial funded by NIH that's going to be happening once we get the grant transferred here from Rutgers. And one of the issues, again, is if we change these things, then, so what? What happens? What else improves? All right, so that's all of my information. And I guess maybe there's time for a question, and if not, we can have somebody else go. But essentially, I'm excited to work with everybody and hope we can develop a center focused on the cognitive neuroscience of schizophrenia. Mm-hmm. And especially, looking at some sensory and perceptual issues in terms of neurodevelopment, predictors of treatment response, predictors of course, and just models of how this all plays into the development of symptoms. All right, so I'm going to stop there.
Ron Wood has a question, Steve. Can you see that in the chat?
I don't see a question from, oh, I see, I have to scroll down. Yeah, that's a good question. Do major tranquilizers thin the retina? Well, there actually is some evidence for that. There's evidence that the antipsychotic drugs actually lead to cortical volume loss, and that's better than the evidence for retinal volume loss. But there is a little indirect evidence that antipsychotic treatment can do that. It's not clear how that works. And one possibility is that by blocking dopamine receptors in the retina, which by the way are found on every type of retinal cell, you essentially stop or slow down activity in those cells. And so some of the cells die off that project to those other cells. But that's far from documented. I see the comment, non-human primate experiments are needed. That's definitely true. So there are medication effects that have to be explored, but I do want to say that while there's no evidence at this point that retinal thinning occurs prior to the onset of illness, there are two very good studies showing that young adults who have no symptoms, who are offspring of people, of parents with either schizophrenia or bipolar disorder, clearly have retinal functional impairments. So there seem to be trait-related aspects of retinal functional impairments, and then there are some state-related as well that you see in patients that you don't see in the high-risk group. And we know from neuro-ophthalmology that you can have about 8 years or so of retinal functional changes on ERG before you notice any retinal thinning on OCT, right? So the cells are kind of getting dysregulated, they're not working correctly years before they actually start to die off. So again, these are all interesting things to study. Another question here is, what's an example of a schizophrenia center that currently exists? There are many of those, Conti centers. Oh, thanks, John. So yeah, that's a good one. Steve Dewhurst. Oh, I started reading Steve's message and then see that it's private. But anyway, I don't think Steve would mind asking if I've spoken with Jesse Shallak. Actually, Jesse and I have just submitted a review paper on retinal biomarkers and neuropsychiatry in general. He's someone I met during my interviews who I'm working with. So yeah, there are a number of people over at the Eye Institute who are interested in these topics.
No, he could do some really nice imaging with you, because they can do that, you know, 2P imaging in human subjects, so that if there were other kinds of data you could pull out that other people won't be able to. That's a unique capacity that we have that nobody else does.
Yeah, it's really amazing seeing the individual red blood cells go through the blood vessels in people's retina. So, all right, well, I feel like I should stop. This has gone on way too long, but I will say I've enjoyed every moment of it. But really, other people do need to talk.
So I'll talk a little bit about some work we've been doing and hope to do in schizophrenia, and in ongoing work that we're doing in healthy subjects, as well as parallel work we're doing in, or hoping to kick off very soon, in autism. And so my title is quite wordy, as you can see, but I'll explain it as I go through. So the basic question that I'm interested in general in our lab is that of perception in kind of naturalistic environments, how we navigate the human world, how we understand what people are saying to us, how we parse scenes, complex scenes that we encounter in daily lives, how we pay attention to a particular person, how we integrate information from someone's face and their voice and block out other speech signals, other music signals, etc., etc. And we think about this in the context of hierarchical processing in the brain. So we think about the fact that there is functional specialization for processing of scenes like this at different levels in different parts of the brain, and the whole process is underpinned by a network of brain areas that interact with each other during daily life. We do some fMRI on this to get at some of these brain areas in terms of how they're involved, particularly in speech and language processing. But our mainstay is actually EEG data, which has its challenges, but also has advantages in terms of obviously temporal resolution and being able to study the processing of complex natural stimuli at fast time scales, which is important for things like speech, for example, which moves quite quickly. And so because of the challenges, EEG has this great temporal resolution, but it's also got poor spatial resolution. So how do we hope to get at this hierarchical processing using a tool as, you know, in some senses as coarse as it is? So the way we do that is we build kind of computational models of how different features of a naturalistic stimulus might predict or relate to different aspects of the EEG data that we record. So an example might be that you can think of a speech signal as being a signal with a lot of acoustics in it. There's frequencies fluctuating and energy changing, but and that's likely to have an impact on, say, early auditory cortex. But you can also think of a speech signal as a sequence of words that carry meaning. And thanks to modern day machine learning and all the rest of it, you can actually quantify the amount of meaning that's associated with words as they stream into your ear. And you can go look for EEG signatures of that. And it turns out that that derives from a very different network of brain areas. And so you can actually get at this hierarchy of neural processing by modeling the data with respect to the different features of the stimulus that you're presenting to somebody. So we've done a bunch of this stuff over the years we've tried to pioneer new approaches for this in the context of multisensory speech using EEG. We've done a lot of work on attention as well. And we've also, as I say, recently branched out into starting to study language and how one might model language and meaning in speech with some reasonable success. And in the past, we had done, prior to the kind of hierarchical stuff, we had done some early perceptual work in autism and schizophrenia. This is when I was a postdoc working with our very own John Foxe. But in the last kind of 10 years, I've branched into this other stuff that I'm talking about now. And the big idea that I want to take forward with this new kind of hierarchical modeling of naturalistic stimuli is the following. The idea is that, our brains, and this harkens back to something Steve actually mentioned about the challenge of thinking about perception as a bottom-up versus top-down process. Well, it's very clear that predictions and top-down do play some kind of role in processing. For example, if I say to you something like, the dentist told me to brush my, well, you now have a very strong prediction about what the next word is going to be, how it's going to sound, et cetera, et cetera. And so the idea is that our perception is actually a marriage, so to speak, of top-down predictions with bottom-up sensory input. And the idea is that in autism and schizophrenia, because of changes in synaptic processing or wiring more generally, there may be a kind of imbalance, so to speak, in the way top-down predictions and bottom-up input are combined to lead to perception. So this can result, for example, in perhaps people with autism not appropriately using top-down predictions of the world and becoming captivated by bottom-up sensory input. And the alternative in schizophrenia might be that patients are overusing their model of the world, over predicting their world and not appropriately using their sensory input, with the result that they may have, for example, hallucinations. And so we have specifically looked at this question: how does some of the EEG models that we've looked at, how are they affected by prediction? We found kind of relationships between top-down predictability of words and how the acoustics are encoded. And we've played around with kind of degraded speech, where we can give people prior information to clue them in as to what they're about to hear, and seen marked changes in the EEG responses that we have acquired. Thankfully, the Simons Foundation recently gave me some funding to try out some of these ideas on predictive speech and language processing in autism. So we're excited to launch that project. It's been delayed by a few months, obviously, because of the current situation, but we're hoping to launch that in September. My proposal was chosen from the U of R to go forward to the Brain Research Foundation this summer to apply for a similar program of research in schizophrenia. And I'm submitting something to the Del Monte Institute later today on that as well. And the whole goal is to use these kind of pilot projects to help launch a robust line of research in both of these populations, and then obviously target NIH for some more substantial funding on these topics. So that's what I wanted to tell you about. And I'm happy to take any questions. I can stop sharing my screen. I can try and stop sharing my screen if I can figure it out.
Any questions for Ed?
Ed, when you're thinking about doing the schizophrenia, like moving these particular techniques into schizophrenia, are you thinking about, patients with an existing diagnosis? Where are you in the sort of predictive piece of it and getting into prodromal populations? Do you have any thoughts about all that?
Yeah, that's a good question. I mean, for the moment, the plan is to try it out because again, I've held off on this work for a couple of years because the EEG measures were so new that I wanted to really, characterize them in healthy populations. And so I think a sensible first step here is to go into people with a diagnosis where we have symptomatology measures on positive and negative symptoms, and we can actually look for correlations between those kinds of symptomatologies and the measures that we're seeing based on particular hypotheses that we have. I think after that, if we have some success there, it would make sense to move towards the prodromes. But I'm happy to hear other thoughts on that as well.
Grand, thanks. So I'm going to talk about, I'm going to try and be brief and talk about some pretty much brand new work. So it'll be very unpolished. So please excuse that. But we've gotten very interested in this particular deletion syndrome, 22q11.2. It's a bit of a mouthful, and that's not helped by. So this is a paper that just came out maybe a month or so ago. I'll be happy to share it with you guys. And 22q11 is also known as, so if you thought that was a mouthful, it's also known as DiGeorge syndrome or Velocardiofacial syndrome. And it's a very interesting...
John, sorry to interrupt. We're just seeing a close-up of your face. I don't know if you're supposed to be showing us something.
Are people seeing the slides? Or
I see the slides.
I think it's because you've pinned my face, Eddie, which I know you like me a lot, but you might want to unpin it. So do you see it now, Ed?
Nope.
Oh, okay. Not sure how to fix that for you. Sorry.
It may just be me. I'll have to suffer through it, unfortunately.
All right. So I'm just so 22q11 is a 3 megabase deletion on chromosome 22. There's a little depiction of it there on the left, and it knocks out one allelic copy of about 36 genes, and there's some interesting genes in there. T-box is one, TBX, COMT is a famous gene that people will know about. It's a dopaminergic gene that's been associated with schizophrenia. And you'd recognize folks with this syndrome. It's rare, but it's not so rare. So you can actually find individuals like this. And one of the characteristics, it's not that everybody with a cleft palate has 22q11 deletion syndrome, but most people with 22q11 deletion syndrome will have this cleft palate phenotype. And and when it's more severe, these are folks who have what we used to call a hair lip. I think it's still called that, and need some surgical intervention. And this particular deletion syndrome is interesting from a number of perspectives, but particularly from the perspective of two syndromes that we care about, and we're talking mostly today about schizophrenia. So what happens with these folks is almost all of them will have an intellectual disability, or as a group, their intellectual attainment is lower. And about a third of them will pick up a diagnosis of an autism spectrum disorder. And another third or so will pick up a diagnosis as they go on in life of psychosis. Specifically, schizophrenia is very common. So about a third of folks, someplace between 26 and 37, 38, depending on which paper you read, will develop schizophrenia. So as of right now, I believe it's the most penetrant, or the highest risk cause of schizophrenia that we know about. And so, of course, that presents it as a very interesting syndromic form or pseudo-syndromic form of schizophrenia, where we understand some of the molecular biology. So that allows us to get a hook into, for example, you could wonder about symptom severity as a function of the functioning copy of a given gene on the preserved allele. And these kinds of questions you can start to leverage into. Okay, so let's go to some of what we do in our lab. Now we do in our lab work that's awfully similar to the work that Steve has done over the years. And Steve and I have sort of gone back and forth with papers about visual sensory processing. And our work has been very supportive, in fact, in terms of finding of each other. And as Eddie alluded to, Ed Lalor did a lot of really cool work back in the days, and still continues to do cool work on these basic electrophysiological phenotypes in schizophrenia. And one of the big sort of famous findings, if you like, in the schizophrenia field, that's now maybe we're 30 years into this, people have used the mismatch negativity. And the mismatch negativity is a simple auditory evoked potential that is generated when an oddball stimulus interrupts a constant streams of stimuli. So for example, if you were sitting in a chair reading a book while we had EEG electrodes on, and we were simply presenting a series of beeps to you, beep, 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 beep. Don't pay attention to them. You wouldn't be paying attention. You could be watching a movie, whatever. So we go beep, 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 boop, beep, 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 boop. Every time that boop kicks off, we would get this nice little well-behaved negativity, the mismatch negativity, that tells us that your brain is representing the regularity of a rhythm and knows when to pick out when that rhythm's been broken in any given way. And one of the more consistent findings in schizophrenia, that these are people with frank schizophrenia, so they've already expressed the disease state, and particularly with regard to duration deviance. So when you use this mismatch negativity design for pitch, I just did beeps and boops, but you could go beep, 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 I'm exaggerating, and just lengthen the duration, and you get a mismatch negativity when that longer tone shows up. So here's a paradigm now, and we're going to take this paradigm where if you were running it in schizophrenia patients, you would absolutely find a deficit in the mismatch negativity. This has been one of the more replicated findings. I'm not going to show you data about that, but we ran it here in folks with a 22q11 deletion syndrome. And so NT here refers to neurotypical controls. And I should mention really quickly, we have a fast rate, so they're going beep, 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 beep. We have a somewhat slower rate, they come every second, beep, 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 beep. And then a much slower rate where they come every 2 seconds, beep, beep, beep. And what happens is as you slow down the rate, the ability to lay down that sensory memory is weakened, and you should get a sort of diminution of the mismatch negativity. That was the thesis. That turns out not to be fully true. So that's all that's happening here. This is the fast rate. Here are the neurotypical controls. This blue trace, the dark blue trace, is the response to the standard, and the dotted trace is the response to the deviant. That's when it got longer. So it takes about 100, they're completely identical, the two tones through 100 milliseconds. And then the system realizes, oh, that one's longer, and it kicks off the mismatch negativity. It's lovely. ERP people get very happy when they see these kinds of things. And if you look at our 22q11 deletion syndrome group, lo and behold, there's no real evidence for any difference. You can see that these two things look pretty damn close to each other. And it doesn't matter what rate we're going at. Here's that slower rate, once a second, nice big mismatch negativity. Here's the even slower one at 2 seconds, really lovely mismatch negativity. If you subtract these waveforms from each other, you can isolate that difference. And so that's what's down here. They're just piled on top of each other, the fast one, the middle one, and the slow one. And these are the neurotypical controls here on the left. And here are the folks with 22q11 deletion syndrome on the right. So no real evidence for a mismatch negativity deficit in these people. However, if we now break that group into those who are not showing psychotic, so these are adults, I should have mentioned that, late teens, early adulthood folks. We have a near 50-50 split in this group between those who have psychotic symptoms and those who don't. And when I say psychotic symptoms here, I don't mean necessarily frank schizophrenia. I mean people who have some soft schizotypal type expressions, but may not be at the level of picking up a full diagnosis of schizophrenia. But now when you break them out like that, what you see is these perfectly healthy mismatch negativities at all these rates for those who don't have psychotic symptoms. And now if you look at the ones who do have psychotic symptoms, you see that the mismatch negativity is diminished significantly. So that's very interesting because that actually aligns very well with results in the schizophrenia populations, where in fact what we find is that in prodromal individuals and in people who have just had their first psychotic episode, there's almost no evidence for mismatch negativity deficits. But it's not until people have the disease, if you want to think of it that way, and have picked up a diagnosis of schizophrenia that you actually see the emergence of the mismatch negativity deficit. So we think, in our hands at least, mismatch negativity is a marker of the disease state and not of the risk for the disease. It's not a good predictor of the disease, but it might be a very useful outcome measure in a clinical trial, for example. So that's mismatch negativity. Now, that paper's already published. I'm going to hopefully be able to grab you for just a couple more minutes and talk about a different paradigm. Now, this is a cognitive paradigm where we're looking in these same folks and we're looking at response inhibition and error processing. So let me describe, now I haven't put up a paradigm slide, but basically what we're doing here is we're presenting pictures to individuals repetitively, and they're completely unique pictures. They're photographs from the IAPS set, if people know what that is. And every now and again, we repeat one. So you just saw a picture of a cowboy, and now we're going to show you another cowboy. And when we show, what we're asking you to do is push the button to every single time you see a novel picture. But when it repeats, don't push the button. Don't push the button. And that's actually devilishly hard to do when these things come quickly. It sounds like a simple task. It's not, because there's this prepotent tendency to push the button on that trial. And then what happens is when you do push the button, you get a series of very nice evoked potentials. And we sort of call them the oh shit potentials. It's like, oh shit, I just pushed the button, shouldn't have pushed the button. And they're known as the ERN, the error-related negativity, which actually comes right out of the anterior cingulate cortex. And then there's a positivity, an error awareness positivity, the PE, that occurs back over parietal cortices. So here's some data from that. And so the first thing to note here, there are two neurotypical groups. Sorry about the confusion on the slide. Sorry, there are two neurotypical groups, and that's because they were age matched. We have a neurotypical group that's age matched to our 22q11 group, and a neurotypical group that's age matched to the schizophrenia group, because the 22q11 group in this case were younger. But what you'll notice here is, here's the ERN. That's what it should look like. The response was just executed here. This is an incorrect response. You should be generating this error-related negativity, and you can see both neurotypical groups kick off a very nice prototypical ERN. Schizophrenia group doesn't, and that's well known. We know that from the literature. But look at these 22q11 folks. They don't kick it off either. And we also find the same with the following positivity, the error positivity. And in this case, what you see is we've broken it out by the 22q11 plus. That means those who have psychotic symptoms. And then the 22q11 negative, those who don't have psychotic symptoms. And we find that no matter which group, they both have the equivalent deficit to people with schizophrenia. So unlike the mismatch negativity, this looks much more like a marker of risk, because it's present in those that don't have expressed symptoms. So it's an interesting dissociation there. These data are literally a handful of days old. We're working on just putting out a paper on that. I have one last slide. Sorry, I know I'm going on longer than I should. Many years ago, in parallel with the work that was going on with Steve Silverstein's in Steve's lab and Pam Butler and so on, we were one of the first groups to show this visual evoked potential deficit. And this is an interesting one. Here, this is from a study almost 15 years ago. And what you see here, the red and yellow trace are folks with schizophrenia. The blue and green trace are neurotypical controls. And what we were showing was early sensory processing deficits in the visual system. And this is possibly one of the most replicated findings in schizophrenia these days. Like I say, many labs have shown it. What I find very cool and interesting is very recent data, literally, I think, again, a month or two old, which is the first time that the full 3 megabase deletion has been recapitulated in a mouse model. So this group came out, I guess, from a Japanese group, because many of the 22q11 deletion syndrome models in mice were much smaller deletions, not the full 3 megabase that we see in the human. So here's the first one that I'm aware of. And there they are showing the visual evoked potential deficit relative to wild type mice. So what's very interesting, again, this is why you would want to study maybe a penetrant or highly penetrant mutation in this group, is because you can recapitulate this in an animal model in ways that you can't with idiopathic schizophrenia. So this now provides us an endophenotype, measures that we can make in humans that we can also make in the mouse model. And that, I think, is where some really interesting future work can go on. So that's me. Thank you very much for indulging me.
Any questions for John? Well, just a comment. I think it's very interesting that, you know, the way you presented those tasks in terms of what may be state marker of psychosis, what may be a trait marker for either schizophrenia or at least some of the genetic component of it. And I think we have potential here to look at combinations of state markers and combinations of trait markers, and how they may reflect various genetic or other biological causes or neurodevelopmental courses and things like that. So that's very exciting that we could put nice batteries together and study things that maybe other centers don't look at.
Right, and I think that's a really key point, that state and trait markers. Because a trait marker, right, if you have a predictor of risk, there's a tendency in the field to conflate those two things. That if you have a predictor of risk, it'll be a good outcome measure in a clinical trial. It won't be. If it's a trait marker, nothing should move it around. It's baked in. And then there's also a tendency in the field to say, oh, if it only shows up in the disease state, it's not interesting because it's not related to risk. But of course, it's profoundly interesting because that's the thing that you can potentially move around with therapeutics. So there is a big difference in a risk marker and an outcome measure, or a measure with high outcome utility.
Right, and I think ultimately you want to answer at least two questions, which is one is, how do these symptoms emerge? And then what's the fundamental predisposition that allows some people to get them some of the time? What's involved in emergence, and then when they're going away, what's happening? That's how you understand what treatment's doing and develop better ones.
I think I'm the outlier here. I think I'm the only animal person presenting, so I just want to introduce myself by way of saying I'm a shrink, and I'm a card-carrying psychiatrist, and my identity is really very centered around that thought. And people with psychosis are actually some of my very favorite patient populations to work with. I've been thinking about this question for a long time. My neuroscience work is in non-human primates. And I'll tell you some story about dopamine and circuits that might underlie psychotic symptoms. So let me just start out with a little preamble to say, I'm totally wearing, my shrink hat right now, and saying that it's important to remember that psychosis is a symptom. And when we say psychosis, we usually mean hallucinations or delusions, which are false fixed beliefs. And to touch on a little bit what, Ed was, I think, alluding to with updating beliefs and, having priors and being able to incorporate new and surprising information into having a more flexible belief system. This is one thing that people with psychosis really are not able to do. But again, it's important to remember that psychosis is not just central to having schizophrenia and other, severe psychiatric disorders, but it's also found in delirium, drug intoxication, some post-anesthetic states, dissociative disorders is very common in them, states of coming and going into sleep, and of course, in dementia, right? So psychosis occurs at any time, and I always tell residents, with enough work, any of us can become psychotic, right? The other interesting thing is dopamine antagonists treat psychosis in all these conditions, right? So we all know that there's treatment-resistant psychosis and schizophrenia, but the rule actually is that dopamine antagonist drugs treat psychosis pretty well. And I guess the question I want to sneak in, and that I've been thinking about for a really long time, is in all the conditions in which we get psychosis, is psychosis really kind of a final little bit of an end stage expression of a disease process, of a brain process? And does it necessarily involve dopamine? So the idea that we want to, my goal is, and I hope to see it in my lifetime, is that we can identify people before they ever get to the stage of having psychosis, because by the time you get psychosis, you're at a final stage, I think, of some of these neurodevelopmental diseases. Anyway, there's plenty of dopamine evidence for dopamine dysregulation in schizophrenia, and I just wanted to hit some of the biggies, which is that we know for many years in humans, striatal hyperdopaminergia is a general finding, both in people with actual schizophrenia, but also in prodromal states and in folks with endotypes, even unaffected siblings, at-risk children. So when you compare folks using PET scanning and provoked dopamine release, people with all these conditions, both risk and the actual disease, actually will have an exuberant efflux of dopamine in very specific places of the striatum. Mouse models are interesting, and it just the 2211 study that John just talked about, I think, is really very interesting. A study last year that really kind of excited me used mouse heterozygotes and showed that they have a lot of the same network and brain cognitive deficits that, you know, mimic schizophrenia and develop by adulthood. And they're fixed by using antipsychotics. But in juveniles, with the deletion, with the hemizygotes, actually treating with dopamine blockers actually prevents the adult phenotype and the outcomes that are seen by adulthood. And I think that was based on mechanisms involving interneurons in the prefrontal cortex. But I think it was a very provocative study. And then in rats, same thing, that animal models that try to model some of the phenotypes of, you know, cognitive deficits that we see in humans, all report excess dopamine firing and increased striatal efflux. And interestingly, adolescent stress actually recapitulates the same dopamine firing pattern. So a very stressed adolescent also has excessive efflux and long-lasting dopamine efflux in the striatum, which I think is interesting. Here's just some of the examples of the studies. Over here on the left, meta-analyses showing that increased dopamine in the limbic associative part of the striatum in many, many studies. Up here in the upper right corner, a study looking at unaffected co-twin children of people with schizophrenia, showing again increased dopamine in the caudate nucleus in those kids compared to control. And then this very interesting study looking at just normal individuals who can successfully update their beliefs in a particular task where they're given surprising new information. And you see that successfully updating information is also associated. This is not dopamine, this is just activity in this limbic region of the striatum. And the people that were successfully able to update information, shown by this sort of signature in the striatum, actually had dopamine levels sort of towards the low end of the spectrum, whereas people that expressed a lot of dopamine in the striatum actually did more poorly on the behavioral updating task. So I think the take home from this was that, you know, maybe this ventral striatal area has something to do with, you know, updating information, and that it's very controlled by dopamine efflux, a very tight regulation of dopamine in that area. So for those of us who went to school several decades ago, here's the old-fashioned dopamine hypothesis, which I think is still, you know, germane, which showed that there is probably too much dopamine in the brains of people with schizophrenia in this structure called the striatum, and too little in the prefrontal cortex. This mediates negative symptoms, the story goes, and that too much dopamine in the striatum actually might be more of a mediator of positive symptoms, psychosis. We don't know how these circuits all talk to each other. And my question going into thinking about that back in the day, in the 90s, was, you know, when people get sick, they actually are usually stressed about something. You know, you see patients come in all the time, something disturbing has happened in their life, somebody rejected them, somebody forgot their birthday, they lost a job, whatever it is, and that's when people get sick. So how does information about the external sensory world and social world get into the dopamine system? And the question I had at that time was, what are the structures that project into the dopamine neurons that aren't just the cortex and the striatum to make dopamine do what it does. We have been focused on looking at a structure called the amygdala. It's here in the medial temporal lobe. And the amygdala is a very interesting structure because it takes in information from the external sensory world, visual information, auditory information, tactile information, auditory and visual, social information, speech, all of that, and it tags it with whether that information is good for us, bad for us, familiar to us, unfamiliar, or whether it's just neutral and just needs to be filtered out, right? So I always give the example of if I'm sitting in a room and people outside the room are talking, usually I filter that information out and it's a nuisance. People with schizophrenia actually tag that information with salience and it becomes threatening to them. So it's inappropriately tagged as something meaningful. To look long ago at whether the amygdala actually projects into the dopamine system, we placed in monkeys substances called retrograde tracers into the dopamine system. And we were able to back label every structure in the brain that has input to the dopamine neurons. And we found that, in fact, this structure called the extended amygdala actually had a huge input to the dopamine neurons. Here's the amygdala down here, but the structure called the extended amygdala, these pink dots are back-labeled cells that projected in. When we put in antegrade tracers into the extended amygdala, these are tracers that feed forward into the all the regions that this area innervates. We saw lots of terminations in the dopamine neuron population. So here's just kind of going from the front to the back of the dopamine midbrain here. And you can see all these little colored speckles are where you see terminals from the extended amygdala projecting in there. Okay, so that sort of set us up to say, okay, the dopamine hypothesis is important in terms of dopamine projecting to striatum and cortex, but it's getting this sort of massive input from the extended amygdala. And moreover, we also showed that there was a input back to the amygdala from the dopamine system. So to me, that sort of sets up sort of a perfect salience loop, right? Information is coming in, those people are talking out in the hall. If it kind of gets mistagged as, you know, maybe in a bit of a paranoid way, or it's perceived as threatening, it happens to be perceived as threatening, it's feeding into the dopamine neurons. And that information is then being fed back to perhaps reinforce that information and also sent out to striatum and cortex. That was work we did a long time ago, and we just sort of set up the basic circuit. And people always said to me, so, you know, but there's no pathophysiologic evidence that the prefrontal, that the amygdala has anything wrong with it in schizophrenia. Is there? And, you know, my answer to that is sort of that it may or it may not. These studies are hard to do, but we certainly know that the prefrontal cortex, the sensory temporal cortex, as well as the hippocampus, all have pathophysiologic changes in different populations with schizophrenia. So information, there's such a massive input here from prefrontal cortex and the sensory world to the amygdala that information that may get miscoded is really being put together there with salience tags, and the opportunity to get mistagged is strong. We then wanted to think about what happens, and this is an anatomic study only, but looking at when stress affects that system, right? So stress is defined as unpredictable, uncontrollable events. And what we knew was that the extended amygdala, this area that projects into the dopamine neurons, has high levels of stress peptides, one of them being corticotropin releasing factor. Whoops, I can't seem to advance this slide. Oh, here we go. So we looked in the midbrain, and sure enough, there was tons of terminals expressing corticotropin releasing hormone, which boosts the effects of the primary transmitters it's expressed with. It actually was also right in the area where the amygdala comes in and projects. So we did that back labeling study all over again. We placed lots of injections of these tracers that both back labeled and forward labeled brain regions into the monkey dopamine system. And what we found was that when we back labeled these extended amygdala structures, that 75 to 80% of the cells all co-contain CRF, this stress peptide that we know does get released each time the animal is stressed. When we looked at those injection sites that back labeled the extended amygdala, we found that they had a very specific set of inputs to the striatum. So this is the striatum moving from the front to the back. And you can see that these very specific regions of the striatum are innervated by dopamine neurons that get input from the extended amygdala. Inputs from cells that don't get extended amygdala inputs have a very different pattern. So we think this is sort of like hot spots of areas where the amygdala may have some influence on dopamine efflux in these areas. And then finally, we actually went to the EM level, and I'll just quickly go past this and not dwell on the data too much, but to ask, you know, what are those CRF contacts actually touching? Are they contacting on dopamine cells? Are they touching on interneurons? And actually what we found is in most cases, CRF terminals contact non-dopaminergic GABA cells, which inhibit dopamine release. That seems counterintuitive, except the terminals from the CRF neurons are inhibitory. So they're actually inhibiting the inhibitor and disinhibiting the dopamine neurons. I just saw a paper from the Malenka lab in mouse showing the exact same setup actually in mice. So the idea is that information coming from the extended amygdala contacts these dopamine subpopulations, which then project to very specific regions of the associative cognitive striatum. And when with stress, when CRF is released, there's probably an gush or an efflux of dopamine to these areas, which actually throws off the signal to noise ratio, causing hyperdopaminergia and affecting function, affecting the ability to update new information. That's a hypothesis. We have a lot of work to do, but we love to lay out the circuitry in normal animals. The conclusions are that the amygdala nigral path is organized to mediate stress effects on very specific dopamine populations, and it can do this because it contains lots of CRF, and that dopamine neurons targeted by these inputs project to striatal regions that actually very closely match the sites of hyperdopaminergia in psychotic individuals, as well as at-risk people. Striatal dopamine must be in tight control to permit cognitive switches and updating, and that the excess dopamine probably contributes noise. As to put it in clinical terms, you know, our miscoded, I guess one thing I wonder is that our miscoded amygdala signals, when information is coded as over-threatening, for example, or one's internal voice is coded as located outside oneself as a hallucination. Are those kinds of signals themselves driving the stress signal, the CRF signal, fueling striatal hyperdopaminergic. So the idea of, the sickness or the symptom fueling itself. And that's all I have to say. We have a lot more work to do. We need to look at this developmentally. We need to overexpress CRF in the extended amygdala and see what happens with dopamine. But we love to lay these things out in the normal individuals first. So that's it. I'll take questions.
Questions for Julie? Well, if there are no questions, let's move on. I did want to say—
I have just a quick one for Julie. It's I mean, maybe sort of a naive view of it. Sort of think of amygdala as a fairly high level, even though it's buried in there in the temporal lobe. It's a fairly high level structure in that it relies on the veracity of information flowing into it from the sensory systems. And it's interesting to think about a front-end sensory system that's unreliable. So here's this information gathering system that's telling you about valence, essentially. And if it can't rely on the sensory inputs, what would be this? I'm sort of thinking about it more from a cognitive psychology perspective, I suppose. But what would a structure that's built to send signals to you about threat do if it didn't have appropriate input? And would you then necessarily expect to find structural deficits in there? There's no necessary reason that would be the case. It's just that functionally it will be doing fairly bizarre things.
Right. No, so I think what you're asking me is, I don't think probably the main place where there's cellular, breakdown or, is the amygdala. I don't. I think since it's a receiver though of every, prefrontal cortex of, sensory modalities, if there's something that is, if it's not coding properly, my guess is you can probably get by with that and do reality testing for a while. But I think that excessive dopamine, every time something surprising or unusual or odd or bizarre happens, right?
Yeah.
Dopamine is being stimulated. So I think the inputs are the things that are probably miswired. I think that amygdala is okay doing its job, but it's getting poor information.
All right, so the title of this presentation, as you can see, is Visual Object Perception as a Window into Schizophrenia and Vice Versa. So I'll just tell you a little bit about myself. I'm assistant professor in the departments of Neuroscience and Psychiatry. I have a little bit of an unusual background because I originally started out aiming to be a philosophy professor. And then I kind of changed gears and got really interested in visual perception non-clinically. And then I joined Steve Silverstein at Rutgers and started investigating perceptual deficits in people with schizophrenia. So the process that has kind of fascinated me in the last number of years is that of visual completion. So this is a canonical kinesis square. You can see that these four Pac-Men are oriented and aligned appropriately so that they form these illusory contours. And the contours in turn encompass this visually completed shape. So this seems like a little bit of a curiosity, but it's actually a really important process that allows us to recover properties of the visual scene, such as object shape, position, and number. So really fundamental properties are recovered through this process. So one of the things that I'm going to be focusing on in the upcoming years is just trying to come up with a unified explanation for the different forms of phenomena that we call completion. So here you'll see pretty readily a kind of salient picture of a rabbit. And even though the entirety of the border of the rabbit is not physically specified, it's pretty obvious to you where the borders are. Now, if you contrast that with this other phenomenon, which is sometimes called completion, this is R.C. James's Dalmatian dog example. you might stare at it and not see anything, but then if I give you some clues about what it is, so okay, here's its head, and it's actually a dog that's kind of putting its head near the ground, then you can kind of visualize the entire kind of outline of the animal. And then yet another example is these kind of Mooney figures, which have been around since the 1950s. Most of us can readily see that this is a face that we're confronted with. And we can also visualize the edge of that, where that face might be. But this seems to be a very different type of completion process than the one on the left with the rabbit. How are these? So we know that these phenomena are similar in that they're both allowing us to kind of see a shape. But we also know that there are important differences because there's conceptual knowledge that plays a critical role in the cases on the right. So are frontal areas like orbital frontal cortex needed when the contours are poorly specified? How and where do these top-down signals and bottom-up areas interact? So for illusory contour formation, we know that certain areas like B1 and B2 and B4, lateral occipital complex, are all really important for allowing us to see these contours. So how do these areas interact with the structures that mediate conceptual knowledge or that carry conceptual knowledge in order for us to seamlessly see objects in these sorts of displays? So I'm just going to show you some recent data from a KO1 study that we're doing with fMRI. I'm not going to get too much into the details just in the interest of time. But in this study, what we did is we had subjects discriminate these visually completed shapes. So in the illusory condition, they would see either a fat shape or a thin shape. And each trial, they have to say which one they saw. And then in the fragmented condition, they saw 4 Pacman that were pointed downward and were oriented slightly to the left or slightly to the right. And they would have to say which one they saw. And so we looked at activation differences between these two conditions. And I'm showing you an inflated cortex here. The cortex is broken into 180 parcels per hemisphere. These parcels were defined by Glasser et al. In 2016 in Nature. And these parcels were defined in terms of functional connectivity, cortical thickness, and myelination. So basically, the advantage of using the parcellation is that you can dramatically reduce the number of multiple comparisons you might want to do for your analysis. So here are the activation differences. The ones that are in color are those that reach significance after FDR correction. So first, I'll point out that V4 and LO areas were highly significant, which is consistent with past research. So that there's more activation in the illusory condition relative to the fragmented. And also, interestingly, certain dorsal areas like IP2 are significant as well. And then finally, an area that I've been interested in, which is orbital frontal cortex, was more activated in the illusory relative to the fragmented. So I should say in passing that most studies have either not looked at OFC or they didn't report anything in OFC, but this could be because of the well-known signal dropout that occurs near the sinuses. So we use multiband resolution, and so we had better signal noise ratio, we believe, than many prior studies, and so we're able to recover this effect. Let's see here. This is an analysis that was done very recently using resting state functional connectivity just to see what information channels exist between these regions, specifically the regions that I just showed you that were significant. And there does seem to be a circuitous kind of route from orbital frontal cortex, specifically parcel 11L, and parts of dorsal lateral prefrontal cortex. So there are 3 different regions, 6R, IFJP, and P946V, showing you these parcels. And then those P946V in turn is correlated, or its resting state data is correlated with IP2, which is a dorsal area that was significant. And finally, IP2 was correlated with V3CD, which is part of lateral occipital cortex. So again, there at least exist these information channels as assessed through multiple regression functional connectivity analysis that suggest that there there's at least a possible way for the orbital frontal cortex to communicate information to dorsolateral areas, then the dorsal areas, and then to mid-level visual areas. So for the clinical strand of my research, I'm looking at this same set of processes, except in the context of schizophrenia. So we have a paper in Journal of Abnormal Psychology that came out in 2019, where subjects did that fat, thin, shape discrimination task that I was just describing. And patients exhibit rather large impairments on the shape discrimination task with Cohen's D of 0.8, even though they perform relatively normally for the control condition that I was describing, where they said left or right. Similarly, there's been work done, I know, by John Foxe and many others, looking at a paradigm where subjects were shown line drawings of actual objects, and these line drawings were fragmented to various extents, and subjects had to identify the object that was depicted. And patients with schizophrenia also show a great deal of difficulty in these recognition tasks. And finally, in the Mooney image tasks, people with schizophrenia are less able to see the faces that are embedded in these stimuli. So a question that has dominated my attention is, does a common region or network underlie these grouping deficits? So, so there's some commonalities obviously between them, in that you're covering object structure, you're seeing the contours, but there's also some differences in that there's a more obvious role for conceptual knowledge in the stimulus types shown in the middle and on the right. Also, for these deficits, these three different types of deficits, is there a role for dorsal regions? It appears that the evidence for that, I think, is pretty good so far. And also, what is the role of certain frontal parietal regions, such as the Perso 11L, which is part of orbital frontal cortex? And then finally, the last point, the last question is, can visual behavior and neural activity deriving from these visual tasks and related tasks reliably distinguish people with and without schizophrenia? So in other words, is there a visual biomarker that tells us whether the person has this condition? So this last point is actually going to be the topic of my R01 application, which I'll be submitting in the fall. And it's going to be more focused on the behavioral work, just showing whether these behavioral tasks can be used as predictors for future psychosis onset. But we'll also be looking at functional connectivity to and from the visual regions to see if those can provide some predictive power as well. So thank you very much for your attention. And if you have any questions or want to follow up, feel free to email me. And then much of this work was supported by my K grant, which is still ongoing.
Thank you, Brian. Hey, can you, we have a question on what is that middle object there?
Let's see here.
It's a watch.
Yeah, this is a line drawing of a watch. So yeah, so people weren't getting perfect performance on that, but in the tasks that were used in these earlier studies, they would provide these line drawing images, and the different fragments of the line drawings would be taken out. And obviously, the more fragmentation, the harder the task would be. And so typically, they would look at to see how much fragmentation could you tolerate while still recognizing the object. And so people with schizophrenia could not tolerate much fragmentation, whereas people who are healthy could tolerate much more fragmentation or much more removal of these line segments.
Well, thank you for having me here today. I'm happy to be here. I'm an assistant professor in the Department of Psychiatry. And I just started up here in January with Steve and Brian Kane. And I wanted to provide a brief overview of my research interest and the type of work I've done as a clinical researcher focused on schizophrenia, and also some of the projects I'm working to develop here at the University of Rochester. I received my Ph.D. in clinical psychology from the University of Pittsburgh. And during that time, I became especially interested in developmental questions related to psychosis. And so as a result, during my postdoc years, I focused my clinical work and also research on the prodromal period of psychosis. In particular, conducting work to characterize risk factors of schizophrenia, such as childhood trauma and cannabis use in young people who have been identified as being at increased clinical risk for developing a psychotic disorder, and looking at associations between those risk factors and attenuated positive symptoms. But I was especially interested in examining indices of dopamine functioning in schizophrenia, so I transitioned to the lab of Anissa Abidargam, and I worked with her group for several years to use PET imaging to examine measures of dopamine dysfunction in schizophrenia, some of what Julie was talking about in terms of those human studies using PET imaging. And we looked at striatal dopamine function using paradigms that allowed us to characterize striatal dopamine release in response to amphetamine, looking at that in people with schizophrenia as well as substance use disorders. And we also used a similar paradigm to characterize cortical and extrastriatal dopamine release in schizophrenia and associations of those measures with clinical and cognitive features of schizophrenia. Most recently, though, I've been working in the lab of with a group of Steve Silverstein. I joined his group at Rutgers, the Division of Schizophrenia Research there, and did work focusing on visual perceptual impairments in schizophrenia, and in particular looking at whether those impairments can be remediated using behavioral interventions. So as Steve was reviewing, schizophrenia is associated with impairments in visual processing across the spectrum, across the whole hierarchy of visual processing. And we were particularly interested in whether a computerized visual training program could be used to remediate those impairments. Those impairments are associated with dysfunction in higher order cognitive processes like visual working memory, also in dysfunction in social cognition processes such as recognizing facial emotion, and even associated with worse functional outcomes. So this is an important treatment target. So we conducted a very small initial randomized controlled trial to evaluate the efficacy of a computerized training program that targeted low and mid-level visual processes.
Clinical trial.
Specifically, this particular intervention targeted contrast sensitivity and perceptual organization in schizophrenia. And the preliminary results we obtained are really promising. They do suggest that this type of intervention can drive improvements in these low and mid-level visual processes. And we're using findings from this initial randomized controlled trial and also just our experience in using this type of intervention with people with schizophrenia to inform the implementation of a larger scale multi-site randomized controlled trial that we're conducting right now in collaboration with Pam Butler. And this larger study is allowing us to more rigorously evaluate the efficacy of this intervention. With the larger sample size, we'll also be able to look at predictors of treatment response to address the question of whether there are particular subgroups of individuals who respond to this type of intervention in particular. And also importantly, whether improvements in these low-level perceptual processes can generalize up to drive improvements in higher order cognitive functions, such as visual working memory and also measures of social cognition. So that is currently in progress. And here at the University of Rochester, I'm also working to develop a new line of research focused on auditory hallucinations in particular. I've had a longstanding interest in positive symptoms of schizophrenia. And through my work, especially with the visual training study, I have been really struck by the number of participants we work with who continue to experience chronic auditory hallucinations, hearing voices despite being on antipsychotic medications and being tried on several different kinds over the years. As Julie noted, a lot of people respond really well to antipsychotic medication, at least in terms of positive symptoms and experiencing a significant reduction or even remission of symptoms from that treatment. But we know that about 25 to 30% of individuals continue to experience persistent auditory hallucinations despite that treatment. And those symptoms can be extremely distressing and disabling when they persist in that way. So there is an urgent need for us to develop novel interventions, especially for those individuals who don't respond adequately to our currently available treatments. So that's what I'm hoping to do. And I was especially motivated by this finding I've noted here on the slide, suggesting that auditory training, similar to the visual remediation intervention we've been examining in the visual modality, that type of behavioral intervention may, for some individuals, target auditory hallucinations and reduce their severity. So I'm very excited to examine the potential for this kind of intervention to perhaps reduce severity of these symptoms, at least in some individuals. The individuals in this study were considered treatment resistant, not responding to antipsychotic medication. So that is something that I'm interested in exploring. But as a first step, I'm looking to examine associations of auditory perceptual processing impairments and auditory hallucinations in schizophrenia to really inform the development of novel interventions, and hoping to actually develop and then test some of those interventions here. And related to that, I've been speaking with Ed Lalor about some of his work and the possibility of using EEG and some of the paradigms he described earlier to characterize the association of speech processing and auditory hallucinations in individuals with schizophrenia. So I'm very excited about that line of work. And I just want to close by saying that a big part of what I've done with Steve's group, and even before that at Columbia during my postdoc years and thereafter, has been to train students and staff in the assessment of symptom severity, and also conducting diagnostic evaluations using measures like the SCID, also for symptom severity, the PANSS and BPRS. And also I conduct a weekly diagnostic consensus meeting where we review all the cases assessed with the SCID to determine the final assigned diagnosis based on DSM-5 criteria. And we'll be getting those going again once we're able to start collecting data again. But I mentioned that because a huge rate limiting factor of conducting this kind of research, as many of you are very aware of, is identifying interested individuals who actually meet diagnostic criteria and our other inclusion-exclusion criteria that we might have for these studies. So we're really hoping to build bridges with other groups here that are conducting research on psychotic disorders or are planning to, because we're hoping that perhaps we can coordinate recruitment efforts and also diagnostic consensus efforts to identify and characterize subjects, but also share subjects across studies, which we've done successfully within our own group for a number of years. So hopefully we'll be able to talk to some of you about that. Thank you.
Well, I guess I'll just say it's interesting work, and I appreciate you putting in those slides at the end about essentially thinking about an assessment core for a center, right? So I mean, you want to do interesting research, but you have to think about how to get it done efficiently, and it would be very helpful to have, obviously, like an imaging group of expertise, the EEG and some other types of methods, but certainly on the recruitment and assessment side, that was a big factor for UCLA and the success of their center, that they had such a great group of people to do all the assessments who are highly reliable with each other. And it's really important. So I'm glad you mentioned it.
Can I ask a question?
Sure,
Steve?
go ahead. Yeah.
Can you hear me? Hi. So I'm listening to all these talks and everything, and I'm thinking about, you know what I'm thinking about? I'm thinking about RDoc. And I'm thinking about, sort of a study. RDoc is this, for people that don't know, this, sort of dimensional kind of way of looking at illness and not placing things in schizophrenia versus not schizophrenia, not in these diagnostic groups. And I'm just throwing it out there. I mean, I think some of these studies on, visual perceptual impairments, and it would be great to, go to a schizophrenia clinic and, recruit new people and program and this and that. But it would also be very helpful, I think, to have, not a non-specialty clinic and get data from those. Maybe you guys know this, clinical stuff better than I do. But I mean, is that what's done, or is that how to think about that?
Well, I'm sure there are a lot of answers to that question. So in the interest of time, I'll try to be brief. We have to still get to David's presentation, but
Oh, I'm sorry. I didn't realize there was another presentation to go. I can wait till the end. I'm sorry.
Well, let me just say for now that, I mean, obviously you're asking a great question, and the whole RDoC, transdiagnostic and dimensional approach is important. I do think you have to look at, the extent to which any of these issues are associated with schizophrenia versus other diagnoses, as well as just psychosis in general.
Right. So that's what I'm thinking about. Yeah.
Yeah, you do have to do that. And, a few years ago when Tom Insel was still head of NIMH and RDoC was kind of a big thing, people were talking like that was going to surpass, or take over as the new way to think about things. But the fact is that there are a number of findings, many really, that where, for example, schizophrenia and bipolar disorder are clearly different. Even though they do share some genes, clinically, when you have people with those diagnoses, they can be very different on things. Even people with schizophrenia and schizoaffective disorder can differ sometimes. So I think an outstanding question that we need to think about related to your question is, what is really related to this diagnosis of schizophrenia, and how can we make that diagnosis better? Or how can we characterize heterogeneity within what we call schizophrenia? And where are the diagnostic boundaries? And what spans those boundaries, whatever they are?
I think for a Conti center, I think that kind of thing would be helpful. We should think about walking up to that because it's a big gooey question. but I think, since we don't have a blood test for schizophrenia yet, you know what I mean? It's interesting. And I often find myself as a clinician thinking about, gee, what is separating this severely dissociative personality person from being psychotic or schizoaffective, or so I think those things are interesting to think about.
Yes, I agree.
Yeah, I would just like to say I completely agree with the points you're making, Julie. And just to make a note relevant to RDoC is a recent PET imaging finding looking at striatal dopamine release and finding that individuals with bipolar disorder during a psychotic episode are showing similar abnormalities as individuals with schizophrenia, and that particular phenotype is very related to positive symptoms in particular. So it's interesting to think about what is shared across these conditions that are having some shared symptoms, but also what you're noting in terms of how, what distinguishes schizophrenia in terms of the
and I guess I was thinking about your study, which is, let's just give these visual perceptual tests to a whole clinic full of people that aren't diagnosed as schizophrenic, right? And see what we pull out. Anyway, I'll shut up now.
I just jump in with a quick observation that it's quite some years ago now, but we did the VEP visual processing study in a cohort of euthymic bipolars and found the deficit in them. And it's interesting, you say, okay, well, what would happen if you went into all these other clinical populations? But we don't find that in autism. We don't find it with aging. I mean, I could go down the list. It's only those two groups that we find this really clear early visual sensory processing deficit. And I think it's particularly interesting in the case of the bipolar individuals because they're all completely remitted when they were being tested.
Okay, thanks, John. There's a lot to pursue here. And I think any center that we would propose would not only have to have interesting methods and all that, but it would certainly have to address some of these questions of heterogeneity, diagnostic boundaries, and then what are the other biological and genetic correlates of some of the performance differences in the groups. But without delaying any further, I want to thank David Dodell-Fetter for waiting. And now we'd like to hear from him about his work with social cognition, imaging, and hear your mind.
Great. Thanks, Steve. Can everyone see my screen? Okay. Great. Okay. Let's see here. Okay, so I'm David Dodell-Fetter. I'm an assistant professor in the Department of Psychology and Neuroscience. I'm a clinical psychologist by training, and I've been here at the U of R since January of 2018. So today I'm going to tell you a little bit about one of the newer and I think more exciting lines of work that we have coming out of our lab, in which we're using real-time fMRI training to try and train volitional control of the theory of mind network in people with schizophrenia. So this is a NARSAD-funded study that's being done in collaboration with Steve Lamberti and tons of help from lots of graduate students, including Arun Ventakaraman, 2 grad students in my lab, Abhishek Saxena, Bridget Shavessel, and my lab manager, Emily Dudek. Okay, so most of my work is motivated by this finding that people with schizophrenia experience profound, persistent, and also somewhat treatment-resistant social difficulties that leave them socially disconnected. Now, this is really important because social connection, or lack thereof, is associated with a wide range of health outcomes. Arguably, the most important health outcomes, ranging from things like well-being and physical illness to mortality. So what this means is that people with schizophrenia are more likely to suffer from the damaging effects of social disconnection. So in a lot of my research, I've tried to address this question of what's contributing to these social deficits in people with schizophrenia, with the idea that some of these processes that are leading to social dysfunction may be amenable to intervention. And in a lot of this prior research I've done, I found that theory of mind, which is the process by which we attribute and reason about the mental states of other people, and its neural bases, which includes bilateral temporoparietal junction and medial prefrontal cortex, plays an important role in how we're able to navigate the social world, and that this network is disrupted in a wide variety of ways in schizophrenia, and also contributes to the types of social difficulties that we see in the illness. Now, we think these data are exciting because they suggest a target for intervention, right? If we could somehow modulate the theory of mind network, we might be able to improve social ability and alleviate the social difficulties that we see in the illness. So our idea behind this study was to see if we can modulate this network by using real-time fMRI neurofeedback. So real-time fMRI is a neuroimaging technique that allows someone to watch neural activity unfold in their own brains over time. And by being able to see neural activity unfold over time in your own brain, it provides an opportunity for someone to become aware of and also control important processes that might be associated with those brain states and which might be impaired in a disorder. So in a typical real-time fMRI experiment, person is put in the scanner, they're asked to regulate neural activity or functional connectivity, something like that. Sometimes they're given explicit instructions for how to do this. Other times it's implicit. The data are acquired and then analyzed in real time. And then participants are provided with some sort of feedback signal, which might look like a smiley face or a number or a thermometer bar or something else like that. And what this feedback does is it tells the participant how well they're regulating and controlling, whatever the brain region is. And so they can learn what what activates this region and what deactivates this region. So there are a decent number of studies that have used real-time fMRI to control a variety of brain regions implicated in a variety of cognitive processes and that are disrupted in a variety of disorders, including many forms of psychiatric illness. In fact, my lab manager, Emily Dudek, and I have been meta-analyzing this literature, and we're finding that by and large, this technique seems to produce the predicted neural change after training. So in the current study, what we're doing is a single-arm proof-of-concept study. We're recruiting 2 groups of people with schizophrenia and people without schizophrenia who are psychiatrically healthy. And they come to the lab for 6 training sessions. So the first session is a clinical behavioral assessment session. The second session is an fMRI session. So we bring them to the scanner and we have them complete well-validated, standardized theory of mind task in the scanner. This allows us to localize in individual participants' brain regions that are recruited for theory of mind. They come back to the fMRI scanner another 3 times to do the actual neurofeedback tasks. And they do 2 types of neurofeedback tasks. So one task is a training task. This is where they perform our task and we actively provide them with neurofeedback as they're doing it. We also have them complete a transfer task. Transfer task is exactly the same, except we don't give them neurofeedback as they're doing the task. The reason or the rationale behind this task is we want to see if they're able to volitionally bring this network online without receiving feedback. So whether it kind of, the training generalizes to a context in which they're not getting this feedback, as they wouldn't get this feedback in their day-to-day lives as they're kind of interacting with other people. And then we bring them back in for a final clinical and behavioral assessment. So just wanted to show you what this training, neurofeedback training paradigm looks like. They basically see alternating blocks where we ask them to either increase or decrease neural activity in regions of the theory of mind network. In terms of the neurofeedback signal, so we give them feedback and specifically median percent signal change from their temporoparietal junction. And again, we're localizing these regions in individual participants using the data from that second fMRI session, or the first fMRI session they do with us. And then we provide them with a feedback value, which is a number scaled from 0 to 100 and a smiley face that is also scaled to that value as well. So again, that's the training task. And the transfer task, which they do every time they come in at the end of the training session, is the same task, except we're not providing them with feedback. Okay, so in the analyses, I'm just going to show you here, basically what I've done is extracted data values from a number of different brain regions that are implicated in theory of mind for this upregulation condition, this downregulation condition, and then I just took the difference, which I'm calling the neurofeedback effect. So basically, we just want to see this value above 0. Okay, so I've rated these data R because they're really preliminary. So we only have data so far from 5 psychiatrically healthy participants and 1 participant with schizophrenia. Unfortunately, we had several patients or several people scheduled around the time that the COVID situation got really bad. So, okay, so in these analyses, I'm just going to show you ROI data from a number of different regions that are implicated in the theory of mind network, including the temporoparietal junction, superior temporal sulcus, different aspects of medial prefrontal cortex, and the precuneus as well. Okay, so here are the data from the training task. So again, on the y-axis, I have this neurofeedback effect. The x-axis, a number of different theory of mind related brain regions. And again, we just want to see this value above 0. And by and large, we're seeing that across the ROIs. There definitely is some variation, though, in terms of how much these ROIs are responding to this neurofeedback training. We also looked at or extracted data just from 2 regions, kind of like a negative control, so left and right frontal eye fields. Again, we're not providing people with feedback from these regions. They're not implicated in the theory of mind network, and so we'd expect no training effect here. And indeed, it looks like we're not seeing that. Here are the data if we break them up by session. Again, they come to the scanner for 3 separate neurofeedback training sessions. And you can see in some brain regions, there seemed to be a pretty clear difference between the neurofeedback effect on the first session versus the last session, in which it's increasing, and in other regions, not so much. And then finally, here are some data from the transfer task. So again, this is the neurofeedback task, except they're not getting any of that neurofeedback. And data here are a little less strong than they are in the training task when they're actually getting the neurofeedback, which is what we would have expected. But by and large, the signal seems to be just above 0, which is positive. We have put 1 participant with schizophrenia through this entire study. Here's the data from that 1 participant. Again, we're seeing positive effect from training. So, neural activity in the temporoparietal junction, precuneus, some temporal pole activity as well. The training data are definitely less robust, but we're seeing some hint of effect in right temporoparietal junction and this ventral medial prefrontal region. Something we're really interested in doing is getting a better sense for what kind of mental processes or psychological strategies, or just what people are thinking about when we're asking them to increase and decrease neural activity as part of this paradigm, and to what extent do certain strategies lead to greater increases or decreases in neural signal than others. And so after every time they come to the scanner, we have them describe to us in really great detail what it was that they were thinking about when we were asking them to either increase or decrease neural activity in their bilateral temporoparietal junction. So here's just an example thing or response that we got from a participant who was telling us what they did when we asked them to increase neural activity during the task. So what we've been doing is we've been taking these responses and putting them through Luke, which is a piece of software that basically characterizes the amount of speed or the amount of text that fits into a variety of syntactic and semantic categories. And just in this analysis, I'm going to show you, I use partial least squares regression, which essentially we're performing a principal components analysis on some of these, syntactic and semantic categories, and we're regressing neural activity during the training task on these different categories. Okay, here's what we found. So the big takeaway here is that, we could kind of summarize the Luke data in terms of two components, a social affiliation and a cognitive processes component, which were explaining, the majority of variance in terms of their responses to this question we were asking, and also the overwhelming majority of variance in neural activity that we saw when we were asking people to upregulate brain activity. So the social affiliation component includes factors like social. So these are words that are getting at social things like talk and daughter, dad, friend, neighbor, and also things getting at affiliation, like whether they said somebody was an ally or friend or whether they were socializing. The second component is getting at cognitive processes, and specifically cognitive processes related to mental state understanding and mental state attribution, like know, think, believe, other things like this. So this is just kind of a nice data-driven way of just showing that people are thinking about kind of things we would expect them to think about when we're asking them to increase neural activity, and that it maps on pretty well to what we know tends to drive neural activity in these theory-defined brain regions and other tasks. That's it. Thanks.
Just want to say, for my part, thanks to all the speakers and to Steve for being such a good MC. I thought it was fantastic, and it's a great way to cap off a long week in the trenches with a real tour de force of fantastic information, and also to be reassured that scientists continue to behave exactly as scientists always do, in interpreting 5 minutes as 25 minutes. That's good. I'm good that we're all exactly as we always were. It's reassuring. But maybe what we might do now, I think, is outside of maybe a very brief discussion, because we've lost half of the folks, is we'll get Elizabeth to pull together a strategy. What was it George Bush Jr. used to call it? Strategery session. And we'll do a little strategery down the road, and that, because I think we might overtax people at this point. What do you think about that for a proposal?
I think that's a good idea. Yeah, we could start fresh and people can think about everything that was talked about today, and we can then come at the strategy task separately. Yeah,
I think a set of fresh minds and just having people chew the cud and digest what they've heard here. There's been great interaction on the chat stream. I've certainly learned a whole bunch of stuff myself. And it's interesting, just mentioning what Julie was saying. They're coming at this with some fresh conceptions about ways that we could leverage into the space, looking at the existing Conti centers and saying, because what you'd want to do is bring something unique to the table, right? There are Conti centers out there. I mean, I actually put up the Columbia one that Dan Javid and Charlie Schroeder have, and we look very like them in some ways, given the kind of span of approaches that we have. So if we were to get into that Conti center world, we're going to have to come with something fresh and new and a little unique, something that only Rochester can do. And I would give that maybe as a final charge to people, like, what did you see? What did you see as a cross-cutting theme? What are the ways in which we could be creative and present a product maybe that would be just unique?
Yeah, well, that all sounds great. And just to get back to, I guess, where we started today, and John, these are things you and I had discussed last week, and that I had mentioned before you came on the call today, which is that we'll have the strategy session especially about a center that we could form, and again, how to make it unique. We also talked about having some type of regular session, maybe every month or two, to go over grant ideas, to review people's data, possibly review journal articles, and kind of just coalesce as a group around ideas that we're interested in. And we also, as part of that, we also wanted to have a way that we could look at each other's grant applications and give each other feedback and kind of accelerate the process of getting grants as a group. And so if anyone does have a grant that they're working on, you know, they should feel free to reach out even now, really, to try to get some feedback. And I'd be happy to look at someone's grant, and I'm sure some of the rest of you would as well. And most people are happy to have other people look at their grant and to kind of get a free review before the NIH people get a look at it.
Exactly, yeah.
So there'll be several follow-up messages coming out about all of these things. And I guess to say, you know, I wish we had more time today, but in a way, the fact that we ran out of time just says something about, I think, the enthusiasm that people have and the disinhibition when it comes to talking about these things. And hopefully that will continue.